My name is Beau. I'm the Chief Scientific Officer for Luminary Therapeutics. Luminary is a recent startup out of Minneapolis. And so one of our big focuses at Luminary is addressing antigen escape. I think we've all seen and have benefited from the remarkable success of CD19 CAR therapy, but one of the problems that remains is the issue of antigen escape. When the cancer cells lose CD19, they are no longer targeted by the T cells. And there have been a number of reasons why it's been difficult to overcome this problem. One of which is the lack of secondary target identification and development, the cost and the complexity of multi or tandem CAR engineering, and then also the heterogeneity of multi-CAR products. And so at Luminary, we've licensed and brought in an innovative strategy to overcome antigen escape. And the technology is a ligand-based CAR based on the BAF ligand, which we view as a next-generation treatment for B-cell malignancies. What's unique about the BAF CAR is that it is able to bind and target 3 receptors, BCMA, the BAF receptor, and TACI. And by targeting multiple receptors, we believe that this makes it very challenging for cancer cells to overcome by losing a single antigen. It also has the benefit as a natural ligand in that it doesn't contain any foreign linker sequence that may be immunogenic. And so one of the encouraging things that we've seen in the field is the prior safety of this general approach, namely using a ligand to target multiple receptors. And there's been a couple of examples here with different ligand-based CARs targeting multiple receptors in early clinical studies that have shown to be safe. And so the concept of using a ligand to bind multiple targets in and of itself is not as promised in terms of safety as has been demonstrated elsewhere. And so just to give you an idea of the breadth of the targets that could be approached using this technology, just looking at a number of malignancies at Luminary, our lead candidate is Mantle Cell lymphoma, but looking at the expression of these 3 receptors, and by and large, many of these cancers express at least 2 of these receptors, if not 3, all 3. On top of the use of a novel CAR platform, we're also utilizing a non-viral genome engineering approach to deliver our CAR, namely the TC Buster Transposon system, which members of the founders at Luminary helped develop before its acquisition by Biotechne, with whom we are partnered to utilize this technology to engineer our BAF CAR T cells. As was explained earlier very nicely, so I won't go into it, transposons offer a number of benefits, the reduced cost, complexity, the reduction in the time to manufacture, which is a big benefit. Of course, there are other benefits as well, including a more preferred integration site profile, et cetera. And so here's an example of data utilizing the TC Buster Transposon system to deliver our BAF CAR. These are primary T cells with a typical sort of outcome that we see, over 65% stable integration of an expression of our BAF CAR. And this is the metrics over 20 independent donor manufacturing runs where we see very high viability after the 7-day manufacturing period, typically greater than 50%, up to even 80% in some cases stable integration, with a copy number that's maintained less than 5. At the bottom, you see a representation of typical memory marker phenotype of the T cells. So one of the interesting things with the transposons as well is the maintenance of a largely central memory or more naive-like phenotype, which is beneficial for persistence and outcome. So here is an example of just the activity against the different independent targets. So here we've engineered a cell line to express each of the individual targets, so BAF receptor, TACI, and BCMA, and we see that the BAF CAR T cells are able to exert cytotoxicity with even a single one of these receptors expressed on the cells. These are 293 T cells. Here again, this is confirmed on the bottom in a different K562 hematopoietic cell line. And so here, functional data against the GECO MCL cell line, looking across a range of effector to target ratios, where we see very nice cytotoxicity early, and that continues over 48 hours, accompanied by the standard degranulation markers. And so this BAF-CAR against an MCL line is very effective, both from a functional standpoint and a cytotoxicity standpoint. This is expanding just again to show the breadth of the potential applications for this, the BAF-CAR-T against other cell lines. So here we also, in addition to the MCL, we have ALL as well as several multiple myeloma lines where we again see very nice cytotoxicity and T-cell activation by the BAF-CAR-Ts. And so to kind of replicate and show the strength of this system and being able to target multiple receptors, here we took the GECO cell line and we used CRISPR to knock out the BAF receptor, TACI, or BAF and TACI together. And what we see is that our BAF-CAR, even in the situation where these antigens, one or even two of these antigens are lost, the BAF-CAR T cells are able to maintain cytotoxicity against these cells. And in vivo efficacy studies, we've seen very, very good performance with the BAF-CAR T cells. These are transposon engineered T cells in a immunodeficient xenograft model produced using the transposon system. And we see a very nice increase in survival in vivo across a very robust efficacy study in vivo in these mice models. So the big thing that we're really interested in is overcoming the CD19 antigen escape in the field. And so we've started to understand how our system will face a situation in which CD19 has been lost. And so one way that we can do this is by actually taking CRISPR and knocking out CD19 in a cell line. And so here we've got a theoretical CD19 positive B cell line and expresses the different receptors. We've knocked out CD19. If you take this population, these GECO cells that express CD19, we use CRISPR to knock out CD19 in half of these cells. We see, and add CD19 CAR T cells, we see an elimination as expected of the CD19 positive targets, but not the CD19 negative population, which persists. However, when we look at our BAF-CAR, we look at cytotoxicity, we see that although the CD19 CAR begins to fail when the population contains a mixture of CD19 negative target cells, our BAF-CAR maintains cytotoxicity even in the absence of CD19. And so we think that this is the first step in a very promising result towards our original hypothesis that this approach could overcome CD19 antigen escape. Finally, in the last slide, I went a little fast here, but I guess I'll be the first talk that I get some questions anyway, which should be great. One of the things we're very interested in is expanding this technology to an allogeneic platform. And the cell type that we're very interested in is the gamma delta T cells for a variety of reasons: their persistence, their homing abilities, their ability to act as antigen-presenting cells. There's kind of a whole host of reasons why they're beneficial in addition to their allocompatibility. And so we've actually started to work up our own novel gamma delta T cell expansion technique that allows for expansion of polyclonal gamma delta T cells, meaning both V delta 1 and V delta 2, which are the common subsets that most groups have worked with in the field. This expansion protocol allows expansion of both of those simultaneously. Utilizing our transposon system, we're able to achieve very efficient engineering, again, in that 40, let's see if I can go back, there we go, in that 40+% range. Something's going on here. There we go. 40+% range, stable integration in these gamma delta T cells. The expansion is extremely robust, as it needs to be to generate an allogeneic product. So here we're seeing typically greater than 1,000 to multi-thousand-fold expansion over the course of the production. And when we engineer these cells with our BAF-CAR, we see that in vitro they perform very similarly to their alpha-beta counterparts. And so just in summary, what we've done at Luminary is develop a next generation CAR therapy for our first indication, which is mantle cell lymphoma. And so far it's shown great promise in terms of efficacy and the ability to overcome antigen escape. We've completed, obviously, the in vivo efficacy studies. We've also completed GLP tox studies with this product and are currently preparing the IND, which we will file before the end of the year, with hopes of starting the initial clinical trial at the beginning of next year. This has all been done utilizing a highly efficient non-viral engineering approach, which we believe will help us be able to reduce the costs, the complexity, improve the safety, and keep the timelines tight for us to be able to manufacture clinical products and bring them into the clinic. I've shown you that the BAF CAR T platform allows for potent activity against multiple receptors in vitro and in vivo. Again, the strength being the ability to overcome antigen escape. Even when one or two of these antigens are lost in that event, these cells are still able to maintain cytotoxicity. And finally, we worked up a new approach for the generation of BAF CAR T cells in gamma delta T cells, which we think is a very promising avenue for the production of an allogeneic off-the-shelf product with unique benefits and obviously broader applicability and all the benefits that come with being able to make a large-scale allogeneic product. And so with there, I'll just leave it here. This is the team at Luminary. We're still relatively small, but we've had a lot of success thus far, and we've got a lot of experience in being able to move products to the clinic in an efficient manner and with non-viral engineering. And so it's very excited to come back next year and talk about us when we're have moved into the clinical phase and have some interesting data to report. So, like I said, I actually kept it on time. So if there's any questions, I'd be happy to answer any questions. Anybody? Nobody's used to having time for questions, I guess. All right. Well, with that, thank you for your time.